Hi everyone, Brenda Gustin here. We're still talking about microbial genetics. We're in the third video, and as you can see, I am in the lecture study guide again. The textbook covers this topic very thoroughly. It's just so much more detail than what I need to do in this course that I thought it was just easier just to give you one diagram and talk about the concepts. So don't hesitate to do some of the reading in the book. There's some really good information there, but I just wanted to use this diagram. So our learning outcome is to compare vertical gene transfer and horizontal gene transfer, and why horizontal transfer is important in bacteria. Now that sounds like a weird fancy genetics thing, but you're quite familiar with vertical gene transfer. That's the transferring of genes from one generation to the next. When any organism reproduces, what they're passing on to their offspring are some of their genes. And so we get genes from our parents, and bacteria get genes from their parents in order to make the next generation. So DNA is copied, it's passed to the next generation, and we call that vertical gene transfer. It's transferred down the line of inheritance. Horizontal gene transfer is something quite kind of different. At one point, I would say it was unique to bacteria, but there's some evidence that some other cells do this too, but certainly not to the extent that it happens in bacteria. And what that is it's transferring genes from individuals of the same generation. And it doesn't even have to be individuals of the same species, but it could be like, oh, you notice I have this really straight hair, and it would be really nice to have curly hair. So I could get some genes from somebody who's got curly hair genes and have them transferred to me, and I could have curly hair. That would be a change acquired by horizontal transfer. Now, we do have some technology that will allow us to transfer genes within a generation. And usually these are pretty fancy technologies that are not used extensively to treat some really serious diseases. But in general, it's not something that we can take advantage of on a routine, but bacteria do. Bacteria are able, many kinds of bacteria, or many bacteria in certain situations, are able to do horizontal gene transfer where they get genes from other organisms and other than their parents. And right here is just some of the three main ways that bacteria can acquire genes. Transduction, conjugation, transformation. You do not need to know the difference among these three. If you took cell biology, you probably would learn that, or genetics, a more advanced course. But in our course, I really just want you to understand that bacteria can acquire new genes from places other than their parents. And so here in transduction, it's just showing you, here's a virus. Sometimes when viruses inject DNA into cells, they actually can pick up a little bit of DNA from the bacterial cell and transfer it to another. So sometimes viruses move pieces of bacterial DNA between bacteria. Here's one. This is the closest thing you'll see to bacterial sects. And this usually this happens in relationship to plasmids. There's certain plasmids that allow bacterial cells to do this, but it's called conjugation, where two bacterial cells that match genetically can build a little bridge between them, and they can actually transfer genes between the bacterial cell. And finally, some bacteria can acquire genes by something called transformation. When bacteria die, there might be some DNA left in the environment, and some bacteria are actually able just to take up that DNA. And so in any of these cases, the bacteria that got the new genes might benefit from the acquisition of these new genes. And again, this is a concept we'll talk about related to antibiotic resistance. So I'm going to stop. Let me just mention that here. Remember we said plasmids are pieces of DNA that typically carry genes that give you special functions. So this is a perfect example of that, where a plasmid's going to carry an antibiotic resistance gene and make this new cell antibiotic resistant. And at that point, I'm going to stop.